Well, thank you, Brooke, for the invitation, and thank you, Emily, for the outstanding organization, and thank you all for coming. It's quite fun to bicycle up here on this beautiful blustery day and get to talk to you about my work, because I think it fits with the spirit of an integrative approach, which I know you all embrace, and I'd be really appreciate hearing your views on what I would like to share with you today. So, like many of us, I'm fascinated with how the brain controls behavior, but also how behavior actually feeds back and influences the brain. I would say that's really what gets my attention. And the particular behavior that I'm interested in is known as vocal learning. And so that's a component of speech and language. And on this slide, I'm just showing you two species of vocal learners, dolphins here and surfers, aka Los Angeles human beings down here. And these are two of the rare animal groups that have the capacity for vocal learning. Actually, among primates, humans are unique in being able to learn their vocalizations, and I'll define what I mean by that for you shortly. But as you can imagine, neither humans nor dolphins are that amenable to bringing them into lab and conducting physiological studies on the brain, and so that's why my group has decided to study songbirds who have this capacity in spades, and much more so than the typical laboratory rat or rodent that you might think somebody on South Campus would be paying attention to. So what do I mean by vocal learning? Well, it's a subcomponent of language, and in an influential 2001 science paper, Chomsky, Hauser, and Fitch acknowledged that it's an important subcomponent, even though it isn't unique to humans. And what am I saying that this is? This is the ability to make new sounds or to mimic another creature using the muscles of vocalization. So you might think that your dog, dogs can do this, but actually dogs and chimpanzees, young ones sound like old ones, except for perhaps a deeper voice because of their increased body size. And what I'm talking about here is also not the idea of learning a word like the word walk, such that you have to spell, do you want to go for W-A-L-K, because otherwise your dog goes and gets the leash off the peg and brings it to you. So I'm really just talking about this capacity to change the sounds that we make using our muscles of phonation, our breath, our larynx, in order to, again, make new sounds, or to mimic others. And so as I've mentioned, among primates, this appears to be limited to humans, and I'll share with you one idea for why that might be. But fortunately for my work, of the 9,000 species of birds on the planet, about half of them are songbirds, and they have this capacity for vocal learning. Now, even though this is not unique to humans, when it goes awry in humans, it can cause a lot of social isolation, and it can be really devastating. And that's articulated here on a little video I'm hoping to play for you of a young girl who's complaining about her speech and language disorder.
Working is terrible because my friends do not understand me a bit. Working is terrible because my friends do not understand me a bit.
So she thinks it's terrible, and she's actually been through a lot of speech therapy in order to be able to articulate that thought to you as well as she can. And so she happens to be a member of what is by now sort of a famous family known as the KE family, whose pedigree I'm showing you here. It's a 4-generational family living in England. And since this slide was formed, the founding mother here has passed on, but there have been additional births of children in this family. And being a neuroscientist, I like to say the KE family is to language as HM is to hippocampal-based learning and memory. So if you remember who HM was, he's this famous case that had intractable epilepsy, and Brenda Milner was very famous for studying him. To remove this epilepsy, surgeons removed a piece of his hippocampus, and thereafter he had absolutely no short-term memory, and he was studied for years and years and years, and really gave us great insight into this type of learning and memory. Well, speech is a different kind of learning. It's a skill learning, and in the brain we think that rather than being associated with the hippocampus, it's associated with other regions such as the basal ganglia, and I'll show you some evidence for that. So this young woman isn't the only person that you can see if you're here in LA. You can Google the KE family. One of the kids named Timmy talks about how he feels being a member of this family. They've been studied for many years, principally by Farena Varga Kadem, and they have deficits in both spoken and written language. They have deficits in the production that you heard, as well as comprehension, and they have deficits in verbal IQ, but they have normal non-verbal IQ. And very importantly, for what I want to get into, it appears that this is a central nervous system problem, because for anything that they need to do to control their face that's innate, like chewing and swallowing and blinking and laughing, they have normal peripheral control. So it appears that this is something going on not between the motor neuron and the muscle, but between the brain and the motor neuron, controlling these regions. Now, if you've been paying attention to this pedigree, you can see that about half the family members are affected. They're the ones shown in red. Also, the females are in circles and the males are in squares, and so it's clearly on a problem with an autosome and not sex-specific. And this pattern of inheritance is consistent with the problem being due to a single gene. So when you think about speech and language, that seems pretty crazy, because it's such a complex behavior that defines us as humans, I would argue, and yet to think that a single gene mutation could give rise to this problem is kind of mind-boggling. So people set out trying to look for this single gene, and meanwhile, people like Dr. Varga Kadem have kept studying this family, and the one trait on which there's no overlap between the affected family member scores and the unaffected family member scores is on the control of the orofacial musculature to make sequential, precise movements. And so in the medical literature, this is referred to as developmental verbal dyspraxia. Okay? Now, from my point of view, this is kind of cool, because this is the type of control that songbirds also have. They have brain regions that enable them to use these muscles to make these ordered sequential sounds, okay? And these people were being imaged in the clinic, and here are just two snapshots of structural MRIs of their brains, here in a frontal view, and here in a transverse view this way. And in yellow is just highlighted the places where there is either more gray matter or less gray matter in the affected family members compared to their unaffected family members, okay? And again, an interesting thing from my point of view, being someone who's studying birdsong, a big hit is in this basal ganglia region that I've alluded to earlier, both in the putamen, where there's more gray matter, and in the caudate of the striatum, where there's less gray matter. And so songbirds actually use specialized basal ganglia region in order to learn their vocalizations. So this is giving me another green light to try to understand what's going on with this family by doing something as unconventional as studying bird brains. So at this point, I just want to point out that I'm happy, because I know this is a nice relaxed format, to take questions of clarification at any time. If I'm not making something clear, please let me know, because probably the person sitting next to you is having the same question and I forgot to say something. So just please let me know. Now, in 2001, Cecilia Lai and Simon Fisher, who were working at the Wellcome Trust Human Genetics in Oxford, England, did indeed locate the single gene that is responsible for this pattern of inheritance. And they discovered that it lay in the coding sequence of a molecule known as FoxP2, which is shorthand for forkhead box P2, okay? And FoxP2, kind of like Gustavo Dudamel, is a master molecule. It's a transcription factor, okay? And so much as Gustavo, shown here with the Children's Orchestra of LA, can elicit from many, many players by having them play more loudly or softly a beautiful concert, FoxP2 can affect the expression of thousands of other genes, either by transcriptionally activating them or by shutting them down. And one of the graduate students in my lab is a big fan of a particular Coen Brothers movie, so he always challenges me to insert the dude in my talk, and so now I've actually been able to do that, and I can move on, okay? So when you think of, isn't it great he's going to be conducting that orchestra at the halftime at the Super Bowl of the, that's so cool. All right, so he's very inspirational, and just like FoxP2, okay. So what is FoxP2? Again, I said it was a transcription factor. How do transcription factors work? They work by scanning along DNA, finding places where there's a little signature that says, hey, sit down and affect my levels of transcription. So they do this by a structural motif in the molecule known as a DNA binding domain, which I'm showing you here, the DNA binding domain for FoxP2. And in yellow, we're just pointing out the place where this single base change was discovered by Simon Fisher. And it causes a change in this amino acid here from an arginine at position 553 to a histidine, okay? And when I fill in the DNA backbone, you can see that that is intimately associated with the DNA. And as you might predict, in vitro studies have shown that this actually affects the ability of FoxP2 to do its job and change gene expression. It is a blustery day. Maybe I should just put it up so it's, or I could. Everybody can still see slides? Okay, I don't have very many color-sensitive slides. All right, so that could be the end of my story, right? So now we know that there's just this single gene base change that causes an amino acid change that affects the ability of a transcription factor to bind to its target sequences, alters the kind of gene transcription that's going on, and ultimately ends up with the KE family phenotype. So we've gone from a base pair to a human language disorder, right? But the funny thing is, FoxP2 isn't just in humans, you know? It's in songbirds, or I wouldn't be talking to you, but it's in organisms that we don't think have vocal learning, like fruit flies, where it was discovered. And FoxP2 isn't only in the brain. In fact, before they discovered this, it was being investigated in the lung, where it helps to organize the epithelial tissue of the lung, along with its closest molecule, FoxP1. So how is it then that this single change in this single molecule can have such a profound and specific lesion on a human trait, right? So I'm going to try and present some evidence for why we think this is possible. And the take-home messages are that we've discovered that the levels of FoxP2 in the brain, not the liver or the gonads or the lung, change during vocalization. So you're thinking about behavior controlling the brain. During vocalization, levels of this master molecule change in the brain. But it doesn't only change during any kind of vocalization. It actually depends on the social milieu. So because I'm talking to you right now and you're my audience, I don't think my FoxP2 levels are changing, but when I was sort of rehearsing the first few lines early on, they might have been going down a little bit. So it depends upon the social context. And when its levels change, so do all the molecules that it regulates. And guess what? Those molecules are different in the brain versus the lung versus the kidney. And so it's regulation by vocalization, by the social context, and I'll even show you some evidence that it's regulated by hearing. That special suite of regulatory things is what makes this such a compelling speech-related molecule. I would argue that if you found another molecule that were regulated in neurons by vocalizing, that were responsive to social context, and that controlled a bunch of specific genes and that were sensitive to hearing, you might have found yourself another molecule that is profoundly linked to speech and language. Now, we learned all of this by studying songbirds, and we think that what we learned from studying these species, such as the zebrafinch pictured here, is relevant to human speech. And that's because they exhibit the relevant trait. They are vocal learners. So in zebrafinches, but not in all 4,500 songbirds, it's a sexually dimorphic trait. And so the male pictured here learns a courtship song from his father that he then sings to the female, and she decides whether or not it's a good courtship song or not. So in other species like duetting wrens, both males and females learn their songs and sing them together. But it's cool for us that only male zebrafinches do this because we can compare the brain of males to females to make insights into how this is occurring. Like humans, they learn through social interactions. So a young male zebrafinch will learn from adult male, usually the one that takes the most care of it. Like humans, young birds learn best during developmental critical periods, after which they're not capable of changing their vocalizations as much. Now that's, in humans, there's a huge span of vocal learning ability after puberty, but I'll just say my husband's Swiss German. I met him when I was 38, and I cannot speak a word of Swiss German. So, you know, critical periods. Like humans, they need to be able to hear in order to learn these vocalizations. Both hear others so they know what they're supposed to sound like. So when my husband says, hui hashli, I say, okay, I know that's Swiss for kitchen cupboard, and it's hui hashli. And he says, no, it's hui hashli. And I say, that's what I said. And he says, no, you said hui hashli. So basically, I can't hear the difference, so I can't make the difference myself. And like humans, they use similar neurocircuitry, such as the basal ganglia loops that I'm going to talk to you a little bit about. And like humans, they learn using similar genetic underpinnings. And this has become even more amazing since the publication of a science journal in 2014, an edition that had 8 papers about birds, and some of them about the similarities between birds and human brains. So I'll show you a little bit of that. All right, so just a little bit of reinforcement of those shared things. Both humans and young birds don't start out making new vocalizations. They make the vocalizations they need to get fed, crying, but they're just sort of listening to the others around them. And some of you may have even done some of these experiments. So there's an early critical period of listening, so one knows what one's supposed to sound like, followed by another critical period where the organism starts trying to make its own new sounds. And in songbirds, they're making these variable sounds that we call subsong. And so these critical periods we refer to as sensory acquisition and sensorimotor learning. And this learning requires being able to hear, as I said. So here's the lovely mellifluous sound of a dad zebrafinch here. Okay, maybe not as great as a nightingale, right? And now here are two sons, one of which was deafened prior to the critical period for sensorimotor learning by pulling out the cochlea, and the other which just underwent a sham control operation where it was anesthetized, and then the membrane over the ear was, there was an incision, and then it was just super glued back up, and so this is a sham control. And now at 60 days, these are both immature birds. This one should improve his song, but his song sounds quite a bit better than that of his deafened brother. So he's well on his way to sounding like his dad, whereas here's his brother. All right, so that's the similar reliance on hearing. And also we said there's a similar use of brain structures. And so here's a schematic of the vocal control nuclei of the songbird brain. And so evolution has really given us birdsong biologists a gift here by clustering all the neurons that are dedicated to vocal learning together such that you can just look at a slice of brain and see in the basal ganglia, boom, those neurons are in the male and they're responsible for vocal learning and they're lacking in the female, and that makes it really helpful for linking brain and behavior. So this is a schematic of a sagittal section. The bird's beak would be here, back of the head was there. And I'm just showing you two cortical regions that get hooked up and then send their axons down to the motor neurons that control the syrinx, which is the muscle that the bird uses to sing. So this is the command pathway for song. And there's this direct connection here between what I would call a premotor or associative motor cortex area and a primary motor cortex. So this would be like layer 5 pyramidal neurons in our motor cortex going down, association cortex going to layer 5 pyramidal neurons in our motor cortex. And then when this innervation happens, that's when the young bird starts practicing its song. So we understand that this is the critical synapse that is required for the babbling of the young bird to begin, okay? But what about these other regions I have here? So in addition, before I get there, I want to say that this direct connection from the primary motor cortex onto motor neurons seems to be a hallmark of vocal learners. So in other non-human primates, these axons would stop somewhere in the brainstem before going down to the motor neurons. And so this is a direct connection in humans and in songbirds that is not present in non-vocal learning primates or non-vocal learning birds like chickens, okay? So we really think that this is a critical pathway for this behavior. That's for the adult performance of the behavior, but what about for the learning? There's a whole other part of the circuit that extends into the anterior part of the brain here. And what this part is required for is for modifying the song during development. So for example, if you lesion some of these regions, the bird can't change its song anymore, all right? And interestingly, this is a cortical basal ganglia thalamocortical loop that, like in humans, is used to control sequentially learned, procedurally learned behaviors. So humans have these cortico-basal ganglia thalamocortical loops, so do songbirds, but again, uniquely in the songbird brain, within each of these regions, there are groups of neurons clustered together that are dedicated for learning and producing song. And we're going to focus on the basal ganglia region because of the KE family and their structural deficits there. And the region of the basal ganglia that the bird uses for its song is got this funny name called area X. I can tell you how it got that name in the question and answer period if you like. So everybody with me so far? Okay. So we wanted to look at FoxP2 in birds, given all of this promising parallels. And so I'm going to show you some images. But we're really going to focus on the basal ganglia. So we're going to take a coronal section here, and you'll be looking at bird brains, bilaterally symmetric bird brains here. All of this region is yellow color-coded for cortex. All of this region is red color-coded for basal ganglia. And then you'll see the bilaterally symmetric song control region known as area X. So the first thing my very first graduate student, Ikuko Teramitsu, did was to try to see where FoxP2 mRNA is in the songbird brain. And so this is a hemicoronal section through the head of a one-day-old zebrafinch hatchling. This is the start of the eye here. The beak would be here. And in black, you're just seeing the signal showing that there's tons and tons of FoxP2 mRNA in this region, which happens to be the basal ganglia. There's also signal in the cortex out here, and this is our sense control. So this is just background levels of signal. And through a collaboration with Dan Geschwind here in the med school, we could examine expression in a 22-week human embryo, and we also saw lots of expression in the caudate and the putamen, which form the striatum. And again, just remember, this is the background signal, so all of this is specific. So this is really giving us a green light to keep looking in the songbird. And these findings are also consistent with the structural abnormalities of the KE family. So you have lots of this transcription factor in the embryonic basal ganglia. If you have a mutation in that transcription factor, then this part of the brain becomes malstructured. Okay? Now, we're really excited about this work because the basal ganglia now, in addition to just FoxP2, the gene expression in area X has been analyzed for not just one gene, but thousands of genes between the songbird and the human brain. And this is this edition of Science that came out in 2014 in December. My songbird colleague, Eric Jarvis, was a co-author on 7 of 8 of these publications. So can you imagine having 7 publications simultaneously in one edition of Science magazine? He got his Howard Hughes renewed shortly after that. So, but the great take-home message for us was when they did these formal comparisons of gene expression, the 2 regions that were the most similar between human and songbird were the genes in the human putamen of the basal ganglia and in the songbird area X, which is what we're studying, and also the human laryngeal cortex. So up here, that place that I said had those direct connections onto the motor neurons that control the muscles had the most similar gene expression to the songbird region that does the same thing with this direct projection. So this is very exciting. So now we know that early in development, we've looked at the brains of zebra finches, and we see that in area X, there's high levels of FoxP2. So if I simplistically graph that as how levels down here are low, levels down here are high, according to this timeline, they're very high in the brain now, at this moment in the bird's life. And if you think about its gene targets, the ones that it's going to activate, their levels should be high, and the ones that it's going to repress, their levels should be low. All right? So that's all interesting if you're a developmental biologist, but I told you I'm really fascinated by behavior. So I wanted to know, what is FoxP2 doing out here during sensorimotor learning, and also what's it doing during the adult use of song? And so we decided to look at that And we remembered that adult zebra finches don't just sing their song over and over again in one social setting. They sing their songs alone as though they're practicing, and then they turn around and sing those same songs to a female. And when they sing to the female, they adopt a little courtship dance. They look at her in a certain way, they hop. So there's some behavioral differences between these two things, even though the song sounds exactly the same to us. So here's a spectrogram of a motif from an adult male when he performs his motif to a female, and then if he practices that same motif alone, and it looks the same to us, and if I played it for you, it would sound the same to us. But interestingly, Neil Hessler and Alison Dope showed that in 1999, there's slight differences. So this performance is slightly more stable. If you record it over and over again, there's less jitter in it. And the neural activity in the brain is actually slightly more stable during this performance, and during the practice, there's a little bit of burstiness in the neural signatures here. And importantly, Sarah Woolley from Alison's lab went on to show that if you play these two songs to a female from a speaker, she can tell the difference, and she prefers the performance. So we decided to look at FoxP2 levels in adult males when they were singing their songs, and to compare that, we looked at them in the brains when they were quiet. So here is another in situ hybridization showing you FoxP2 mRNA levels in the brain of a non-singing male. And we've just used two different probes to assess this, essentially replicating our findings in one study. And we're plotting the level of the signal here relative to the level of the signal outside of area X, and there's just a slight enhancement within area X. Now, what happens when the bird sings looks pretty much the same, okay? So maybe you can see area X a little bit in this one section, but we have about 20 sections per bird and about 6 birds per group, and so there's statistically no difference here, and it looks pretty similar to what we've seen in the hatchling bird. Dan?
Sorry, if this is the technical detail I want to skip by. How do you know gene expression while the animal is singing or not? I mean,
we would love to be able to track gene expression in a living animal, but you can't yet. And so these animals woke up and either were quiet for 2 hours in the morning or sang for 2 hours, and then we killed them.
Okay, got it. That's fair.
Yeah. Anything else? All right. Well, now you probably know what's coming next. What if instead of performing the song to the female, the male practices his song by himself? All of the same song sounds the same to us, but guess what? FoxP2 levels are dramatically different, and now you can really see the outline of all those basal ganglia neurons in area X dedicated to vocal learning because they show up. And FoxP2 levels dramatically drop precisely in the song control neurons and precisely when the bird is practicing. So even though the song looks the same and sounds the same to us, FoxP2 levels are very different, and they're responsive to the social milieu. And this relationship is very profound in sensorimotor learning, such that Ikuko could just take these few birds and show that the more that they sang, which is here on the x-axis, the lower their FoxP2 levels are. These are in hearing birds. Interestingly, this relationship does not emerge in birds who cannot hear themselves. And this was our first link of the molecule to hearing that, in fact, anybody had made. So to summarize then, things that I told you were take-home messages, we think we can see hints of a post-organizational role for FoxP2 in learned vocalizations beyond just the structural formation of brain structures in embryogenesis. We've seen that specifically within the basal ganglia song control nucleus, FoxP2 levels decrease during vocal practice, and this regulation depends upon singing, social context, and hearing. And so in a sense, the birds are self-regulating their own levels of FoxP2, and maybe these cycles these behavior-linked cycles are really important for this behavior. So here's my cycling graph here. Instead of just these static levels in early embryogenesis and hatchling life, we can tell that during sensorimotor learning, FoxP2 levels are going up and down, and so are all the different molecules that they are controlling. All right? So what are those molecules? We'd really like to know. And to get at that, we did the following experiment, which is just using the bird's own behavior to modulate levels of FoxP2. We know if he sings a lot, they're going to be low. If he doesn't sing anything, they're going to be high. And seeing how many other genes change similarly along with FoxP2. So this is our experimental design. On a given morning, 24 birds woke up, the lights came on. We recorded whether they sang or not and how much they sang. At 2 hours, their FoxP2 levels are either going to be low if they sang a lot or high if they sang nothing. And then we micropunch their brain for the song control region, and we also micropunch an adjacent region that contains exact same cell types, and then look at the transcriptional profiles of these regions. And so we actually used one of the first cDNA chips arrays that was available for zebra finches. So we took the mRNA from those regions and hybridized them to an array, and we got what genes are up, what genes are down, and all these 24 birds, and we used systems analytic technique, unbiased, known as weighted gene co-expression network analysis, to build a gene network to cluster together groups of genes whose levels are changing in concert. We built that network. This is only a third of it. At the same time, we took the recorded song and we analyzed the song for all sorts of features like how crisp is it, how much is there, how loud is it. And then in the end, we sought to correlate any of these gene expression modules with any of these features. And the key here is that all of this was done completely blind to any of this. So anything that comes out is from an unsupervised analysis. Now, just off the top of the experiment, you can do something much less fancy than WGCNA. You can just look at the interarray correlation for the, now I think it's 54-ish arrays that we had put brain mRNAs against. And if we cluster them, and this is a dendrogram much like a family tree, the further down here things are together, the more similar they are, the longer it takes to get over to another one, the less similar they are. Reassuringly, all the area X samples cluster together in their gene expression profiles, and all the basal ganglia regions outside of area X cluster, giving us good feeling that brain region is a good determinant of gene expression patterns, right? So that's reassuring. Now, if we just look at the area X samples and we cluster those, or look at the correlation between them, we find that all the birds that did not sing at all cluster together with the gene expression profiles from birds that sang the least and are very far away from the birds that sang the most. So this is telling us now that singing behavior is a powerful modulator of gene expression in area X. Okay? What about FoxP2? When we did this, we had no idea whether there were any probes on the array for FoxP2. It turns out that there were 12 probes on the array for FoxP2, and in each and every single one, the more a bird sang, shown here, the bird numbers are, these are individual birds, the lower the levels of FoxP2 expression. So this really shows us that this is not a freak phenomenon. So the initial statistics on this work were that after correction for false discovery rate, within area X, there were 1,372 genes that were modulated or correlated with singing, and 1,837 genes that were correlated with how much of the singing. So this is yes, no singing. This is yes, singing, how much. And in the outlying region, there were absolutely no genes that were correlated with singing. All right. So what are these genes? What are they doing? Well, we went ahead and, as I said, my second graduate student, Austin Hilliard, used WGCNA to cluster these genes. So these are the actual probes on the array. This is, again, a dendrogram. This is 1 minus topological overlap. So the further down, the more similar they are. And then Steve Horvath here at UCLA has a program where you can use a dynamic tree cutting algorithm to prune this dendrogram, looking for patterns in the DNA, and plop these groups down into what they call gene modules. And now you have these groups of co-regulated genes, and you can ask, now, are these correlated at all to any of the traits that we were interested in? So you're unblinding the little man in my prior schematic. So this is a heat map, and red means highly positively correlated, and green means highly negatively correlated. And I'll just give you the stats here. We ended up with 3 different modules, the yellow module, the tan module, and the green module, wherein these genes were highly significantly correlated with either positively to whether the bird sang or not, how much it sang, or negatively. And where is FoxP2 in all of this? It's exactly where it should be. It's very negatively regulated by singing. So it's here in this green module. And here's another gene that people had been examining and already knew was positively regulated by singing. And sure enough, it falls right here in this yellow module. And here's a gene that we don't think has anything to do with singing. And sure enough, there's absolutely no correlation with these other song-regulated genes. Okay? So we've got clusters of genes or modules that are changing depending upon singing. Now, how unique are these to area X? So I'm just replotting this area X data over here. You've already seen it. This is the dendrogram. These are the 3 modules. Over here was another part of the network that came out. These genes were not regulated by singing, but when we used different databases out there, we found that they were enriched for cell types, okay? So these are the aqua and red gene modules that are enriched in basal ganglia cell types, all right? So let's go and look at the region outside of area X, because we have those arrays as well, and we can build a dendrogram for those. So this is the network for the ventral striatal pallidum. And if instead of giving these genes the colors they would have in the ventral striatal pallidum, we paint them with the colors that they would have had in area X, we see this pattern. And very reassuringly, we see preservation of this aqua module and this red module. And that makes sense because these are also striatal pallidal neurons. So it's made up of similar cell types. But we see absolutely no preservation of these song modules, all right? And going further, we can compare plain old levels of expression of genes in area X versus levels of expression of genes in the outlying area. And there's the same number of genes represented in this graph as in this graph, but they all line up on unity here. So the overall levels of expression are very similar in the 2 brain regions. And what's actually very different is how they are co-regulated with one another, how they go up and down together rather than individually. And so what we're saying then is connectivity, or the co-expression levels of genes, more than the expression levels themselves, appears to be defining the molecular specificity of a brain region dedicated to vocal learning. So these are the discoveries I previously told you about, and now I can start to fill in how FoxP2 can have such specific actions. It's likely due to the fact that it's uniquely regulated in vocal control regions by hearing, social interactions, and vocalizing. And when it changes, it's part of a molecular microcircuit that is specific to area X relative to other areas. And so we think that alongside of synaptic and anatomical microcircuitry, this molecular microcircuitry contributes to the functional specificity of brain regions. Now I just want to show you a few more slides that describe our functional intervention into this circuit. If we're hypothesizing that these cycles of up and down regulation of the circuit are important, how can we prove that? What we would want to do then is actually break the cycle by pegging FoxP2 levels high during this time and breaking its regulation of all these other gene targets. So to do that, we developed an adeno-associated virus. My graduate student John Heston did this, that when he injected it into the brain, now you can see the mRNA for normal levels of FoxP2, but you can also see this enhanced level of mRNA expression for the injection that he gave, and it's pretty well targeted to area X, which is shown here in just a crescent section of the brain. When we look for the levels of reporter expression, we see that we get them pretty well localized, and we validated this in a way that I can share with you if you have questions about it. But suffice to say, we're capable of overexpressing both the mRNA and the protein in the bird brain. And so now for his experiment, he would take a tutor that had a couple of sons, and in one of the sons, he would inject this adeno-associated virus that would overexpress our reporter gene GFP on both sides of the brain. And in another son, he would overexpress FoxP2, okay? And he did that. Here is the surgery, and then he removes the tutor so that the bird has had enough time to learn its tutor song, and now the birds live by themselves, and we record them, or with a female, and see how well their song is progressing. So that data is shown here, just for one tutor and 3 pupils. So here's what the tutor song sounds like. See if I can play that. And in his GFP expressing son, pretty good. In the 2 FoxP2 overexpressing sons, okay, so that's qualitative data. Here's some quantitative data from 8 GFP expressing birds and 9 FoxP2 overexpressing birds. And when we compare the similarity to the tutor, we have a significant decrease in the quality of the learning. And just for full disclosure, we never went back and made sure that, for example, this bird did indeed have a precise surgical injection to area X. So we were able to pull out significance despite the noise of the actual neurosurgery itself. And when we analyzed when these changes start coming on board, almost as soon as we record our birds, so they have this surgery at, a little bit before the onset of sensorimotor learning, they start babbling right at that innervation at about 35 days. When we get their records at 50 that we can actually evaluate that they have motifs, we're already seeing that the GFP injected birds are doing much better than the FoxP2 injected birds. So, yes?
Clarification. The virally induced overexpression is persistent throughout many, many days. Is that right?
Yes, yes, and we did confirm that, yes, yes.
I was wondering how exactly you quantify the similarities with tutor.
Yeah, so there's a couple of methods out there. One which we've actually developed, but we didn't use because we hadn't developed it yet, called VOICE, Vocal Inventory Clustering Engine. But we actually used a program called Sound Analysis Pro that's available from Ofer Chernichovsky's group, and it's what most people use to quantify zebrafinch song. So it looks at things like entropy, frequency modulation, goodness of pitch over many variables. And for the talks like this, we just sort of break it down into sort of a global analysis. For example, I can tell you when we did some kind of syntax, I shouldn't say syntax, syllable sequencing analysis, we did not see any differences. So all of the differences we're seeing are phonological differences. Okay. All right, so is it that these birds can't change their song at all? So in, I think, my last data slide, I'm just going to half slide, show you this same graph here of these birds, but instead of comparing their songs to the tutor, we compare their own songs to their own adult final song, so the bird's own song. So compare the song here to the song here, right? And when you do that, you see that the birds are changing their song over development with a very similar developmental trajectory to the GFP expressing birds. So the birds are actually capable of changing their song, they just aren't capable of making a good match to their tutor's song. Now, we weren't the only group looking at FoxP2 in birds. We overexpressed it. A different group, led by Constance Scharf at the Max Planck in Berlin, knocked it down. And very interestingly, we come up with really similar phenotypes for either keeping it high or keeping it low. In both, we have incomplete motif copying, poor syllable copying, feature-specific errors in the duration and entropy of these syllables, an early emergence of the deficits, no effect on sequence, and no difference in this trajectory of change. Really importantly, both of these, all of these outcomes are very different than what would happen if you went in and simply lesioned area X. So this isn't just, oh, you're mucking with area X, this is what you get. And so we think the parsimonious interpretation of this is that the thing that's in common in both our studies is that we've disrupted this dynamic regulation of FoxP2, and we think that it's necessary for the fine-tuning that underlies vocal learning. And so we've also been looking at this in adult birds, where my postdoc Nancy Day has been overexpressing FoxP2 in birds that she deafens. Here's just a snapshot of a bird's motif over time. It stays really stable for a long time beforehand. Then if she puts in GFP, it stays stable. And then if she deafens the bird slowly, like in humans, the song degrades. Really paradoxically, if she overexpresses FoxP2 instead of GFP, the song degrades even faster. So this is another link to hearing. You're seeing a much earlier onset of deafening-induced deterioration, again suggesting that the cycles of FoxP2 that we know are disrupted in deafened birds are important for the maintenance of song. And this is just data from 4 of her birds, 2 that got the GFP and their songs were deafened, and this is how they degraded over time, and 2 that got the FoxP2, and you can see this abrupt decrease. So the take-home messages are, FoxP2 is vocally, socially, and hearing regulated. It's co-regulated with different suites of genes in adjacent brain regions made up of the same cell types. And we think it's these features that lend it this specificity such that the KE family doesn't have a problem with their manual dexterity, but they have a problem with their orofacial motor control. And perhaps behavior-driven cycling of key transcription factors is an evolutionary phenomenon that may underlie types of procedural learning. So with that, I just show a picture of my group outside of the Terasaki Life Sciences building. Ikuko is now an assistant professor at UNC, so she's not here. John is going to go get a postdoc at San Diego pretty soon. Nancy just came back from field studies in Ecuador, recording in vivo from duetting wrens. So I'm thrilled about that. It's not my work, but I let her go. And she came back. And so, and I want to thank some of my collaborators here at UCLA, some of my funding, and for your attention, and I'm excited to hear your thoughts. Yes.
So, trying to extend some of the parallels with humans, and I realize that you work on birds. The birds have affordances in terms of what you can do with them, humans don't have.
Go for it.
So the first is, I mean, it's not surprising that FoxP2, well, maybe in retrospect it's not surprising, I don't know, but, you know, fruit flies have it, zebra finches have it, humans have it, that there would be affordances in the gene that it would, convergent evolution would produce parallels in vocal learning. That doesn't sort of blow me away. What does blow me away is the parallels in the anatomical structuring, right? Because the common ancestor of birds and humans is pretty darn old, and that the same sort of modular organization of the brain would result in the, would be put together in the same ways in order to arrive at vocal learning seems pretty surprising, since normally you would expect, analogies to just have superficial resemblance and not such
Right,
proximate resemblance.
right.
So one, I'm curious whether you have any ideas as to why that is. And the second is, since, at least not until the technology changes, we're not going to see FoxP2 expression modulation in human brains as a function of performance in social context. I wonder if behaviorally there are parallels that could be explored. So is it the case, for example, that vocal performance during private rehearsal and during public performance is acoustically different, and that listeners can detect the difference, given that in the birds, vocals seem to be.
Well, I appreciate both of those questions. First, with regard to the brain structures, I think it's super cool. I do not have any scientific knowledge, but I have anecdotal knowledge that the same relationship regards the opposable thumb. So in creatures that have an opposable thumb, like raccoons, there's this direct connection between the primary motor cortex onto the motor neurons that enable that. Okay, so another convergent specialization that has an anatomical underpinning. And Eric Jarvis, who did those 7 papers in Science magazine, has been talking to us because one of the molecules that he found to be uniquely regulated in the human primary motor cortex of the larynx and the bird is called SLIT1, and it regulates axonal guidance. And so they're developing adeno-associated viruses to either express this in a non-vocal learning, probably ringdove, or to knock it down in a vocal learning bird to see if they can actually create a Frankenbird, right, that can have some of the features. Now, I wouldn't say all, right, because you don't just have that vocal motor backbone, you have the basal ganglia loop, and that connectivity, likewise, is not fully functional, for example, even in female zebra finches, right? Another approach has been to expose zebra finches to certain hormones, because it's a sex behavior, and Art Arnold, Barney Schlinger here, have tried really hard to make a female zebra finch become a male zebra finch by exposing them to different hormones. And that's been very difficult because it appears that a lot of this exposure has to be really high within the brain concentrations to get these anatomical specializations. I will say that parrots and hummingbirds are in different orders than songbirds, and they're separated in the avian tree by birds that aren't vocal learners, but they are vocal learners. And if you use this immediate early gene that I alluded to earlier, referred to as EGR1 or ZENK, and its levels go up when the bird sings, you can see that the levels are also regulated in these different birds. And the thinking is that it evolved 3 times in the avian lineage. So in all 3 cases, you can make the argument that the structural arrangement is really similar. So that's what I can say to that. And just remind me the second part of your question.
So since we can't, at least not for the time being, measure FoxP2
Right.
Modulation in human brains as a function of performance in social context, can you look at the acoustic and subjective aspects? And that if the finch finches can tell the difference between a practice song, if the females can differentiate between a practice and a performance song, right?
Right.
And when you look carefully, you see acoustic differences. Is the same true in humans with regard to, say,
Right.
I don't know, some aspect of performance?
Yeah. So again, I don't work with humans, but there are studies by people like Ben Olveski at Harvard, who works in birdsong, pairing up with another researcher who works in humans, and the whole effort is on whether the jitter, the behavioral jitter, is noise in the system or whether it's adaptive. So can the central nervous system not perfectly produce a basketball free throw over and over again? If you could just nail it every single time, you'd be, the most coveted player, and yet we can't, right? And so is this noise a bug or a feature of the nervous system? And what their work has shown is that this variability is adaptive, that if they look at humans learning different movement trajectories of hands, those that show some of the more variable movements in the beginning end up becoming the masters more quickly. You can imagine if you're trying to reach a certain peak performance and you try to approach it, I'm talking sort of, learning theory, incrementally like this, it might take longer than if you're exploring motor space. So the idea is that this variability is actually adaptive for producing the perfect sound. Females can tell the difference. I guess I would think about virtuoso instrument players or something like that. And I know that there are studies that find that if there's a robotic performance of a piano concerto versus a master, that the slight variability that the master introduces is deemed more beautiful, actually. And even work here with Reggie Edgerton in my own department, helping to train people with severed spinal cords to walk. They put them on a treadmill, and they started out using robots to move their feet on there. And they found that the ability to recover that motion was much worse than when they used humans moving their feet. So the nervous system, I think, is using this slight variability. And I think that it's appreciated by the female zebra finch. I know I said that it's much more crystalline when he performs to her, but I think that's probably a part of him actually having practiced it as well. So understanding exactly the valence needs to be worked out, but there's certainly hints that the variability is adaptive. And a final say on that, my colleagues at UCSF, Michael Brainerd, and others at MIT, Michael Fee, have been using an auditory playback where the bird, well, in the extreme case that we're actually adapting in our lab, the bird wears little headphones, okay? And you track the bird's song, right? And you track the fundamental frequency of its syllable. And there's a natural level of variation in that. And you can get triggered feedback to play the bird back white noise, or you can overplay another version of its note as a way of punishment in real time. And when you do this, you can get the bird to start singing at a lower fundamental frequency if you punish the higher, or vice versa, if you punish the lower, it'll sing higher. So this shows in principle that the variability can be used to learn to avoid this noxious stimulus, and that the bird might be paying attention to this. It's not very ethological, but it's cute to see the birds with the headphones on. Yes, Brooke.
So I think this follows up a little bit on that, but I'm just wondering what the role of competition then. So when these birds were performing, like I assume it was just the male and the female together, right? There weren't other males around. But I'm wondering, particularly in species where, in natural settings,
Yeah.
be competing, how that.
Yeah. Yeah, it's a great question. So the way these female preference studies is done, and I wasn't very clear about this, is you record the male when he's singing to her, and then you record him when he's singing alone. And now you take the female and put her in an arena by herself, and you play her the male's song from one speaker when he was singing and the other, and you see which one she spends or which she calls back to more, right? So we haven't looked at males competing with each other under those conditions. It would be really cool to have, well, I actually have this data, and I'll be able to weigh in on this. So we injected another virus into area X that expresses a designer receptor that's exclusively activated by a designer drug. Does anybody know about these dreads? Right, so these are G protein coupled receptors that are in our brains that usually dopamine or something binds, and it causes the neurons either to be slightly hyperpolarized or slightly stimulated, right? Now Brian Roth at UNC has engineered them that they aren't responding to any endogenous drug, they're responding to this new drug. So you can stick them in, they don't do anything, they're not activated, and then you can feed the bird this drug and you can dampen or excite those neurons. And when we do that, the song slightly gets more variable or less variable. So we can have the male bird with his normal song when he's singing to the female, his normal practice song, and then his dreaded, more stable song, and then his dreaded less, and we can ask her those questions. Female preference, maybe some of you know, is super tricky. And so, you know, sometimes you put a bird in a cage and it just wants to go hang out on this side, and you have no reason why, and you know. So, but that would be a way not of testing between males, different males, but of testing within a single male a full range of his variability. If you're asking more about the ethological meaning of these songs, work by Steve Nowicki at Duke suggests that the brain is still developing post-hatch in these birds as they're being nurtured by their parents, and such that the quality of the song reflects the fitness or the nourishment that the male received during this time. And so they've been doing some studies, and I don't know what the recent outcomes are, where they food-deprived females to see if she can tell still if she's food deprived, you know, if she's nutritionally deprived, and also if there's an impact on the quality of the male's song. In general, you can go to certain species, and the more, I mean, there's lots of birds that, unlike the zebrafinch, that sing multiple types of songs. So we have Bengalese finches in our lab, but there's, you know, I mean, the amazing lyrebird, right? So there's species out there, and the more, this work has been done by Elliot Brenowitz at Washington. He has shown that the greater, you can see a correlation between the greater the repertoire, the larger some of these brain structures actually are. So, yeah. Yeah.
Do the male finches invest anything besides speaking their offspring, their sons and daughters?
Yeah, I mean, I think so. So if I know how you guys think about this, the male really, he's a big provisioner of the young, and he builds the nest and uses that and courts the female, and then he provisions the young. Sometimes we rear birds as isolates, so we remove the male, and so in that case we give a female nanny to try to help make sure that they're going to survive. They're not very territorial. That's part of the reason why we like to have them in the lab, because they love to be close together in big flocks. So there's not a lot of mate guarding or territorial behavior. So does that answer what you were interested in? Okay. Yeah.
Is it the same kind of gendered asymmetry in those other lineages that developed over in other lineages?
Great question. Now you've got me. Do hummingbird females and males, is it sexually dimorphic in hummingbirds? I do know in parrots, so it's fun in parrots because the female does learn. And actually, if you have, this is work with Tim Wright at New Mexico State. Females sort of get together, budgies at least in the lab. I mean, he studies parrots in the wild, but in the lab he just looks at budgies, and the females coalesce on sort of a dialect, and when the male is introduced, he changes his song to try to match hers, which is kind of fun. And they've been testing kind of sort of a passport hypothesis of what that might mean. We've looked at FoxP2 levels in his parrots. They are lifelong learners, and what we found is that FoxP2 levels are constitutively down-regulated in the part of the brain of the parrot that we think is the same as area X. So we never see it go up. And so, of course, the hypothesis would be maybe that's part of how come they're lifelong learners and able to change their songs throughout their lives. And so we're gearing up to overexpress our virus in those parrots and see if that prevents them from being able to learn new dialects.
There's no cycling though?
That's what we haven't been able to catch any kind of cycling yet.
I'd love to hear the story behind how it became called area.
Oh, that's a great one, because not too many people know. So it was Fernando Nottebohm. I don't know if you know him. He was at the Rockefeller. He's one of the godfathers of birdsong. He was a disciple of Peter Marler. And so he was at the Rockefeller, and his first graduate student was Art Arnold, who's here, a distinguished professor at UCLA for, 30 years. And they were trying to figure out, because they're interested in neuroendocrinology, whether there were neurons in the brain that were sensitive to sex steroids. So they took hot, radioactive estrogen, or maybe it was testosterone, and injected into the bird, and then waited. And of course, that's going to bind in the brain to the steroid receptors in the brain. And then they did this in a zebrafinch female and a zebrafinch male. And then they sacrificed the bird, sectioned their brains, put a film on. So now you've got an autoradiogram. And there were 3 blobs in the male brain that weren't in the female brain. And Fernando took out a Sharpie and wrote X, Y, Z. And Y and Z's names have been rechanged, but area X has always remained area X. Isn't that weird? When I was as a new faculty sometimes at the Society for Neuroscience, they'll have you host the Songbird social. That's like your big, networking. And so I had to do it when I was new faculty. And so I had a competition to rename Area X. And I got to give out prizes based on that. We came up with some Area 41, some funny names, yeah.
I'm sorry I missed the first few minutes, so I don't know if we touched on this, but does a finch learn equally well from a foster male as a biological father?
That's a great question, yeah.
Once a finch is an adult and has learned its song, if that finch is introduced to another area where the bird songs are slightly different, can that bird adapt and change that song?
Right. So for the zebra finches, they're closed-ended learners. There are other songbirds like canaries, for example, that will learn their songs and change their songs seasonally. But in zebra finches, once they're adults, the only thing we've been able to show that their songs change is by deafening them. So their songs are really stable. The parrots, which are not songbirds, like I just said, are capable of learning throughout their lives. So they will change their songs. A fun thing we can do in the lab is we can cross-foster birds. So I mentioned that we have Bengalese finches. There's a beautiful story there about these finches that were indigenous to Bengal and were picked up by Japanese bird fanciers and bred for their plumage and not for their song. And they evolved a much more complex song. Now, instead of referring to them as white-rumped munyas in the wild, these 300 years worth of breeding we have what are called Bengalese finches, and their songs are much more complex in terms of the syllables because you've basically released the male from the pressure to keep his song somewhat recognizable in the field. And now, in these bird cages, she's only got a choice of who's there, right? And so the female preference for the more elaborate song has evolved. And if you take that song and you play the white Ramunya song to a naive white Ramunya, she prefers the Bengalese song. Well, we use these Bengalese finches to tutor our zebra finches. So after they've pair bonded and had one clutch on their own, the next time she lays eggs, we swap out her eggs and throw in the zebra finch eggs. And the zebra finch will learn to the best of its, oral motor ability, the Bengalese finch song. And that's very convenient because then you can parse out, what is genetically inherited aspects of song versus what has to have been learned, right? So that's some of the fun games we can play. Yeah.
So I think this might be sort of putting together your question with Leah's question earlier about the tutors, and maybe this is more of a behavioral question than might be your
Behavioral?
area. Yeah.
Okay.
That might be your area of expertise, but so I'm wondering about how much of this is paternal investment versus just sort of passive learning from the tutor. So that's why I think putting together the fosterage idea, and then, because extra-pair copulations are so frequent in a lot of bird species, whether if you were not to cross-foster across species, but just pair them with another zebra finch who was clearly not the biological father, whether they would teach less if in fact it's teaching that's going on, or if this is more passive learning.
Yeah, I don't know. That's a great point. Is it passive learning, or is the male actively teaching? Yeah, and I'm not sure people are looking at that. I know there's some studies kind of looking at single moms and stress, but I don't. In terms of the father investment, I'm not sure.
Do the adult males vocalize more when they're near offspring than when they're not? Because that would differentiate.
I don't rear them that way. I rear them in a house together, and so it's a family cage. And we do have big aviaries, but I don't keep track of that. So that would be a good way of getting at that. Yeah.
Do they have high rates of extra pair paternity?
I don't think so. Now, I know that chickadees do. I know that you can overplay a male chickadee's song in the wild and demote him in the social scale, and then his female will go off and seek extra pair copulations. Zebra finches are pretty monogamous, and I can say that we are always trying to get the male to sing, and we want his song a good recording, so we take him and put him in the recording chamber. And if he's in the middle of a clutch with a female, it's harder to get him to sing. He's like, he's more interested in being back. That's the anecdotal, So they don't, when you're trying to get your lab going, you have to really work at getting pairs to bond with one another. They don't just go, yay, So there's something. And there have been studies looking a little bit at female choice. Apparently, she cares about the levels of carotenoids in his cheek as a fitness reporter for his immune status. And she weirdly prefers males that look like her dad. You guys, I don't understand that, but those are some of the studies I'm aware of. But I don't know about the paternal investment.
Do you know if it might be possible to use this richer genetic profile of the gene's development of vocal learning to maybe extrapolate an emergent state phylogenetically of this trait in bird species? Of course, I ask because I don't know if that might be possible to do in humans as well for vocal learning.
Well, I'm trying to recruit a graduate student who is actually going to look at the genomes of these white Ramunyas versus these Bengalese finches to try to look at genomic signatures like that. And then my addition to it would be to try to look at transcriptomic changes, because people very often just sacrifice their animals without paying any attention to what the bird or animal was doing right beforehand. And our data would show that you're just really going to get a different answer if you're not actually paying attention to this. You know, in those science papers where they looked at the gene expression in deceased humans, for example, we have no idea what those humans were doing before they died and the state of the brain and the levels of vocalizing. And I would think that those things would influence those. But I don't know if that's a full answer to your question. But another approach that's being undertaken is to try to breed species that don't, like a non-vocal learning species with a vocal learning species, and then examine genes that are in the progeny that segregate with the vocal trait or not. But that stuff is, you know, just in its infancy. That's work in Michael Brainerd's lab at UCSF. Well, thank you all very much. These are great questions.